cholesterol actually holds the phospholipids back a bit, keeping them from getting too liquidy. And when it gets cold, it gets in the way, preventing them from packing together too tightly. It's the perfect temperature manager.
All right, so the membrane is this fluid, temperature-controlled structure. We've got that down. Now let's get to the main event. Let's see how that structure allows it to do its most important job, controlling traffic. It's time to see the gatekeeper in action.
So, broadly speaking, there are two main ways things get across the membrane. Passive transport costs the cell zero energy. It's like rolling downhill, from a high concentration to a low one. Active transport, on the other hand, is the workout. The cell has to spend energy, usually a molecule called ATP, to push things uphill against their natural flow.
The most basic kind of passive transport is just diffusion. You've seen this before. Imagine dropping food coloring in a glass of water. The molecules just naturally spread out until they're evenly mixed. Small things like oxygen and carbon dioxide can do this right through the membrane. No help needed.
But what about stuff that's too big or has an electrical charge? They can't just squeeze through that oily middle layer. For them, the cell uses something called facilitated diffusion. It's still passive, still going downhill with the flow, but it uses special proteins that act like selective tunnels or shuttles. A perfect example is something called an aquaporin, a channel just for water that lets it zip through way faster than it could on its own.
Okay, but what happens when the cell needs to bring something in that's already rare outside, or it needs to pump out waste that's already crowded out there? It can't just rely on things floating in. It has to fight against that natural downhill flow. It has to spend some energy.
And here is the superstar of active transport, the sodium-potassium pump. It's constantly at work, so let's walk through it. First, 3 sodium ions from inside the cell latch onto the pump. Then an ATP molecule comes along and gives the pump a little packet of energy. That energy forces the pump to change its shape, and poof, it spits the sodium ions out of the cell. Now, with its new shape, 2 potassium ions from outside can bind. That causes the energy packet to pop off, and the pump snaps back to its original shape, releasing the potassium inside the cell. Now here's the crucial part. For every single cycle, the cell spends 1 ATP to pump 3 positive charges out and only bring 2 positive charges in. This tiny imbalance, happening millions of times, builds up an actual electrical voltage across the membrane. And that voltage is vital for things like sending nerve signals.
Controlling ions is a huge deal, but what about controlling the most important molecule of them all, water? This is where we have to talk about osmosis, and it is absolutely make or break for a cell's survival.
To really get osmosis, you need this one concept, tonicity. It sounds complicated, but it's really just a way to compare the concentration of a solution to the concentration inside a cell. It tells you whether that solution will make a cell gain or lose water.